recording. Okay, so what this second part of chapter 3 is going to do is it's going to go over the prokaryotic cell. And this is the deal. If you read the chapter in the book goes over both eukaryotic cells and prokaryotic cells. I don't bother going over the eukaryotic cell because I assume you already know it. This is the cell you studied in biology 1406. So this is the cell that has the nucleus and the mitochondria and the endoplasmic reticulum and the Golgi. So if you've forgotten some of these organelles and what they do, you can go back and read about them in the book. What I am going to do is I'm going to focus on the prokaryotic cell, and I'm going to contrast and compare prokaryotic cells and eukaryotic cells every time we introduce a feature of prokaryotic cells. Okay, so no mitosis, definitely no. There's no need for mitosis. Yep. Yeah, I'm definitely not going over mitosis. I'd rather spend the time understanding the prokaryotic cell. So first thing about the prokaryotic cell is that the shape is very important. It's one of the criteria for classification. Shape and gram reaction is the first thing we do when we're trying to classify a bacteria. So what kinds of shapes can we find in a bacteria? Well, we have the round shape, which we call cocci. We have the elongated shape, which we call rods. You can also call rods bacillus, but if you call them bacillus, make sure you don't capitalize the word bacillus, because bacillus with a capital is the name of a genus. So don't do that if you're talking about the shape. Okay, so there's a little cocci, and you can see that cocci looks like a little pearl necklace, little round circles. That's another cocci right there. These are rods. Okay. Some bacteria have other kinds of shapes. We have the spiral shape, which is a curved rod. The C shape or the comma shape, which is called vibrio. And so this is a little coccobacillus. A coccobacillus is a tiny little rod, which when you look at it, you're going to say, oh, this is a cocci. Well, it's not a cocci. It's a tiny little rod. The way you can tell them apart from a real cocci is that when you have more than one side to side, or in a line, you're not going to have a pearl necklace. You're going to have a line. So see, can you see a line here? You can see this line right there. So you have a longer rod. That's what you end up with. Whereas if you had a cocci, you would have a pearl necklace. So that's how they would be you'd be able to tell them apart. This is a vibrio, a curved rod. I know it's kind of hard to see, but it's just a little curve of a rod. Yeah, actually, let me show it to you really quick. Okay, notice here that you have a cocci in a chain. And okay, so you have a cocci in a chain, and when you look at it, it looks like a pearl necklace. When they are in a chain, these are dots in a chain, and dots in a chain will look like a little pearl necklace. If we have a rod in a chain, we're going to have a straight line. We're not going to have that pearl necklace appearance. And yeah, it's not easy. And you kind of have to look around, because if you look down here, you're going to say, oh, well, that looks like a straight line to me. Yes, it does. But this doesn't look like a straight line, and that doesn't look like a straight line, and neither does that. So you have to look around, and if most of them do not look like a straight line, then they're cocci, a chain of coccis. Now take a look at what's the difference between SPS and SPP. I think the SPP is just a misspelling. It should be species. No, that should have been just one SP. That's a species. So when we write down a Streptococcus, a species, so that means, does the textbook uses 2 P's or 1 P? I don't remember. Maybe I'm the one who's wrong. The textbook is right. So if the textbook uses 2 P's, then that's what I should be using. Both. Oh, really? Huh. Interesting. Okay. I don't know. My guess, I'll look it up. I've never noticed. My initial guess would be SPP would be a species, meaning all species, versus SP, meaning that one particular species. That would be my guess. But I'll look into it. I've never paid that much attention. Good for you guys for paying attention. In this case, if SPP means all species, that would make sense because all the species in this Streptococcus genus are going to be cocci. So there are no exceptions. We're talking about every single species. So let's say for now that that's what that means. And then I'll investigate further. I was going to show, yeah, we were going for these coccobacillus. Okay, so when you look here, that's a straight line. That's more than one cell in a line. This is more than one cell in a line. And you can see how you have these straight lines. Look at that little straight line. When you have those straight lines, you know that's a rod, a little bitty rod, but it's a rod. So this is not a cocci. And yeah, it is sometimes hard to see. Vibrio, we're not going to have vibrios, so don't worry about vibrios. It's not a common shape. Spirillums, those are the curved ones. Spirochetes are even more curved than spirilla. This one is a different kind of shape. This is a rod. However, when you look at these, some of these rods are big and some are small. This is all the same species. It's all the same genus, forget the species. It's all the same genus. So it all should look the same, but it doesn't. So we call these pleomorphic. When you have, this is all the same bacteria, and yet you have some fat little rods, you have some tall, long rods, you have some, small, medium shapes. So these are all different sizes. That's not usual. Typically, when you have a rod, they all are more or less the same size. Here, they're all different sizes. They're pleomorphic. That is typical of a genus called Corynebacterium. And then we have the weird shapes like a star shape and square shape, which are different kinds of bacteria that have been found. So that's the shape. Don't confuse shape with arrangement. Arrangement is the grouping that the bacteria take as they divide. So some bacteria, when they divide, they may take on specific groupings. So again, SPP, all of the species of the genus Streptococcus are going to be chains. So they're going to be cocci in chains. And that's the arrangement. So this is shape. Chain is the arrangement. And if this is a gram stain, then you can say this is a gram-positive cocci in chains. So you're given gram reaction, shape, and arrangement. This is a cluster of cocci. So these little packages, sometimes are described as clusters of grape. So all members of the Staphylococcus genus have these cluster arrangement. Tetrads are packages of 4. So these little square packages of 4 cocci. So the shape is cocci. Shape cocci, arrangement tetrads. These are diplus. So 2, you can see here 2, 2, 2. So little groups of 2, diplococci in this case. So these are doubles, and the shape is cocci. So this is shape. This is arrangement. And a lot of the arrangements are especially important in cocci. Okay, so this is the pleomorphic rod. The arrangement of these pleomorphic rods is very unique. Notice that some of the cells, grant you not all of them, but some of them arrange themselves side by side, like that. Sometimes one in the top. Look at these. That's typical of Pernobacteria. No, all this information. Okay, so yes, somebody's asking that they already turned in lab 2 and they didn't know all this information. Should I go back and rename? Yes, go back, fix your lab report, and turn it in again. The lab reports, I have set them to admit as many documents as you want. So resend it, and I will grade the last, the one that was uploaded last. So absolutely, do that, most definitely. Because that one is not due until Saturday. So yeah, you have time. Go ahead and do that, most definitely. Okay, so this is called a Palisades arrangement. A nickname was Chinese letters because some of them do look like a little bit like Chinese letters. Like, do take a look at that, or this stuff right here. Okay, it's Palisades, so that side-by-side arrangement. Unique. This is a unique arrangement to the genus Corynebacterium. So some of these arrangements are unique to specific genera. Okay. So some bacteria can make sticky stuff that is secreted to the outside, and with that sticky film, they can stick to surfaces or stick to each other, and that creates something called biofilms, which are communities of bacteria that live together in their natural environment. They're sticking to each other, sticking to surfaces, and the importance of biofilms is that they're very difficult to get rid of. The bacteria are very thick. Yes, dextran comes from biofilms. Actually, dextran makes biofilms possible, exactly. So some bacteria are able to secrete this sticky sugar called dextran, and with that, they stick to each other, they stick to surfaces, and they have layers and layers of bacteria that are very difficult to penetrate by antibiotics or disinfectants. So it's a protective layer. The plaque and teeth are biofilms. So yeah, dextran is one of the substances that bacteria make to create biofilms. So shape of a bacteria called pneumococcal pneumonia. That should be pretty self-explanatory. Okay. All right, so one of the reasons why we're going to spend so much time looking at the prokaryotic cell and contrasting prokaryotic cells to eukaryotic cells is because first of all, because it allows you to grow the bacteria, if that's what you want to do. It also will allow you to prevent their growth, if that's what you want to do. It also allows you to selectively destroy one type of cell, let's say the bacteria, over another, let's say the human cell. So it allows you to create good drugs. A good drug, a good antibiotic, is one that harms the bacteria and it doesn't harm the host, because it is going to target something that is unique in the bacteria and is not present in the host. So we're looking for the differences between them and us so that we can target what is unique to them and we don't have, that way cannot harm us. Killing bacteria is very easy. It's the easiest thing in the world. The hard thing to do is to kill the bacteria without killing the patient. That's the hard thing. So we need to find drugs that kill the bacteria without harming the patient. That is called selective toxicity. So we need drugs that have selective toxicity. So this ability of a drug to cause harm to the infectious cell without harming the host. In order to do that, you need to target something that is unique to the bacteria and essential for the bacteria. So you can kill it without harming the host. So as we go through these structures, we're going to ask ourselves, could this be a good target for an antibiotic? Is this something that our cells have? And how similar are this structure in our cells versus the prokaryotic cell. So these are the things we're going to be looking at: cell membranes, cell walls, the capsules and slime layers, and then inside the cell, there isn't much inside a bacterial cell. The DNA, that's basically, ribosomes. All right. So that's just pictures of prokaryotic cells. This is a list of all the structures of prokaryotic cells. Okay, we start with the cell membrane, and here's off the bat, same composition as eukaryotic cell membrane. So that right there would that be a good target? It is telling us off the bat, our cell membranes are basically the same. We both have phospholipid bilayer in our cell membranes. Yeah, there are some proteins there, true, and those proteins may be unique, but when it comes to what's making the bulk of the cell membrane, it's basically the same. We've seen cell membranes in chapter 2. They're made of phospholipids with the hydrophilic heads, the hydrophobic tails. Okay. So we saw these already. One little difference between their cell membrane and our cell membrane. Our cell membrane has sterols. Prokaryotic cell membranes do not have sterols, except for Mycoplasma bacteria. Sterols are these fats, lipids, that are positioned in between phospholipids in order to give the cell membrane more stability, because the cell membrane is fluid, is an oil. Okay. Functions of the cell membrane: movement of things in and out. Okay. The movement of things in and out can be by diffusion, whether it's simple or facilitated, or by active transport. Active transport requires energy. This right here is unique to bacteria. The cell membrane of bacteria is the site of ATP synthesis, of energy synthesis. In the eukaryotic cell, the site of ATP synthesis is the mitochondria. Bacteria don't have mitochondria. They're going to make ATP in their cell membrane. Okay. Let's go over transport, and we'll stop there. So simple diffusion is the movement of substances following a concentration gradient. You should have seen these in biology 1406. So the movement will be from an area of high concentration to an area of low concentration. To cross a cell membrane, which is made of phospholipids, the substance that is going to diffuse has to be small, and it has to be hydrophobic. So if it is small, just because it's small, its chances of going through are pretty good. So something like oxygen, it's very small, and it should be able to go through the cell membrane following a concentration gradient, movement by diffusion. CO2, which is a waste product of the cell, it's very small. It should be able to leave the cell by diffusion. If the molecule is larger and it is hydrophobic, it should be able to go through, because the cell membrane is made of lipids and it allows lipids through. If the molecule is hydrophilic, so let's say something that has charges, like a protein, okay, like a protein that has charges, it will not be able to go through. This is hydrophilic. It has to be hydrophobic. Water is small. However, it is charged. It's water. So it's not going to go through the membrane easily. Most cells are going to have little pores called aquaporins, and water is going to go through it. So even though the pictures don't show that, in reality, water is not going to necessarily move easily through the cell membrane. It will move, but it will have to be through a little pores. Nonetheless, the diffusion of water in and out of a cell is called osmosis, and it does follow a concentration gradient. So if a cell, which in this case is this yellow thing here, is immersed in a solution that is very concentrated, meaning it has a lot of solutes and not very much free water. The key word is free. So these could have water, but the water is being trapped by the solute, so that the free water molecules are not very many compared to the inside. The inside has lots of free water molecules and fewer solutes. The solution outside compared to the one inside is hypertonic. So this is hypertonic in comparison with the inside, which is hypotonic. So a hypertonic solution has very little free water compared to a hypotonic solution, which has more free water. So in those situations, there is more free water inside the cell that will be moved out compared to free water outside the cell, which is not very much of it is gonna move in. So there's gonna be a net loss of water. So a cell in a hypertonic solution is gonna lose water and shrink. A cell in a hypotonic solution, more water outside, more free water outside than inside. The free water will go inside the cell. The cell is going to gain water, and it may burst or be damaged. So you should know what happens to cells in a hypertonic solution versus a hypotonic solution. So again, simple diffusion of water is called osmosis. Now, this is the deal with prokaryotes. Prokaryotes have a cell wall. Cell walls protect prokaryotes from hypotonic environments. So if this cell is a bacteria, it will have a cell wall. And as the cell membrane swells and becomes bigger and bigger and bigger, and make it another color, I'll make it blue. So the cell membrane continues to grow. It's not going to explode because the cell wall will stop it from growing too big and exploding. So cell walls are protective in hypotonic environments. The minute you remove the cell wall from a bacteria and you put it in a hypotonic environment, it will explode. So the cell wall prevents this rupture of the bacteria in a hypotonic environment. Most bacteria live in hypotonic environments. So they've adapted. They're adapted to hypotonic environments. So do you want to harm a bacteria? Give it a hypertonic environment. They have no protection from a hypertonic environment. The cell will shrink and die. We've been doing this for thousands of years without even knowing what we were doing. We were taking foods and loading them with salt. And we found out that when we did that, the foods didn't go bad so quickly. That was because this was a hypertonic environment. Any bacteria trying to colonize the food was going to shrink and die. We've been making jellies filled with sugars, creating a hypertonic environment to preserve foods. So food preservation for thousands of years have been used in hypertonic environments to kill the bacteria without even realizing that that's what we were doing. Okay, so do know the role of the cell wall of bacteria in hypotonic environments. And out of the two environments, for a bacteria, this is the worst one, the hypertonic one, because they have no protection. Okay, got that already. Yeah, we talked about that too. The cell membrane is the site of ATP synthesis. Eukaryotes is the mitochondria. All right. Let's stop here. We have a little bit more transport to do, but that's not very much. So we can pick it up from here next time. From okay, that's good. 147. Is that for aerobic and anaerobic bacteria? Yes, it is for both. Aerobic and anaerobic has to do with oxygen, not with osmosis. So both aerobic and anaerobic bacteria will be susceptible to hypotonic environments. There are, yeah, so that has no, oh, the ATP. Oh, okay. Oh, that's okay. Now, maybe, I see what you're saying. Okay, so is aerobic and anaerobic, are they making ATP in the cell membrane? Maybe. Definitely aerobic. Aerobic bacteria make ATP in the cell membrane, no question about it. Anaerobic, it depends on how it's making ATP. If the bacteria is making ATP by an anaerobic electron transport chain, then the answer is yes. If the bacteria is making ATP by fermentation, the answer is no. So it depends on what the anaerobic bacteria are doing. An anaerobic bacteria could have an electron transport chain that doesn't use oxygen, in which case it's using, it's using the cell membrane. Or anaerobic bacteria could be fermenting, in which case it's not it's not in the cell membrane. That's the cytoplasm. So that's chapter 4 and chapter 6 that we're going to deal with that. So the answer is maybe. In an aerobic, definitely yes. In an anaerobic, maybe, depending on the bacteria and depending what the bacteria is doing. So yeah, absolutely. That's actually a good question. Welcome. Okay, so let's stop there. Lab on Monday, and I guess Monday we'll tackle lab 3, I think it is, which is actually an easy lab. So we'll tackle lab 3. And yeah, definitely, lab Monday, 8 to 8:30, yes. That's this later times. Oh, whatever I have. What did we do last time? Did we do 8:30? I can't remember. Lab. Let me look at what we have right now. Okay, last time was 8:30. Okay, then let's stick it to 8:30. Let's stick to 8:30. It will be a short meeting. Yeah, tonight ended up being 8:30, and I may have to change it to 8:30 because I have that biology 2402, which is going longer every time. So we'll go ahead and stick to 8:30. I'll send out an email, change it to 8:30. 8:30 seems to work better. Otherwise, I'm going to be, we're going to be having the same problem as we had tonight. Oh, I'm recording all these. That's okay. The same problem as we had